Welcome to the Wednesday afternoon lecture series. This is, of course, the NIH-wide forum for the most distinguished scientists in biomedical research. I'm Mike Leonardo. I'm from the National Institute of Allergy and Infectious Diseases and hosting our delightful speaker today. I'm thrilled to introduce her. Her name is Hao Wu, and she's from this field of structural biology and visiting from Harvard Medical School. Dr. Hao Wu is the Asa and Patricia Springer Professor of Structural Biology and a Professor of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. She's also the Associate Director of the Program in Cellular and Molecular Medicine at the Boston Children's Hospital. And she's world-renowned for her work on molecular mechanisms of instructional insights into signal transduction in cell death and immune pathways. So she earned her bachelor's equivalent from Peking University in Beijing, China, and then went to Peking Union Medical College. While at medical college, which I assume was to become a doctor, she participated in laboratory research in immunology and, as rumor would have it, became enamored by basic research, and instead of finishing up her MD, decided instead to emigrate to the US, where she pursued a Ph.D. in Purdue University from the renowned X-ray crystallographer Michael Rossman. Her postdoc was at Columbia University with another renowned structural biologist, Wayne Hendrickson, and in 1997 became a faculty member at the Weill Cornell College of Medicine in New York City. She was there for 15 years, reached the rank of professor, and then moved her lab northward to Harvard and the Boston Children's Hospital. Among her many, many landmark discoveries, and I can say this from personal experience because we've had the good fortune to collaborate with Hao on a couple of projects, has been identifying the oligomeric supramolecular assemblies of various proteins within the cell that carry out important functions in the systems of cell death and immunity, particularly the death domain proteins, which we had a strong interest in from a public health and from a disease standpoint. I would say she rewrote the textbooks when she discovered the structures underlying the mechanism of the inflammasome activation through these higher order assemblies, a theme that goes on throughout her work and on many different important aspects of the immune system. She found that these death domain complexes have a helical symmetry, which kind of harkens back to work by Francis Crick and other outstanding biologists. And they have unusual stoichiometries. I guess this was what surprised me the most. 5 to 7 for the PIDD-RAIDD complex for caspase 2 activation. In the pitosome, the 5-5 fast-FAD complex and caspase 8 activation by the TNF receptor family member FAS, and 6-4-4 for MyD88 IRAK4 and IRAK2 complex in the midosome. So these are very unusual structures. I don't know if those are Fibonacci numbers or why nature evolved to have those numbers, but the bottom line is that it was very surprising and came directly from her structural insights. Her lab has also done other things, beta amyloid structures, which are, of course, the hallmark of Alzheimer's disease. She's also become an outstanding cryo-EM scientist in addition to her work in crystallography, so now she's imaging even larger structures in the cell. And I just want to point out, this is just a snapshot of her remarkable career. She's had many, many honors for the contributions she's made. She was a recipient of the Mayor's Award of New York City. I assume you got that from Mike Bloomberg, which is pretty impressive. This year, she's the Thermo Fisher Meritorious Career Awardee from the American Association of Immunologists. She's also elected as a Fellow of the American Association for the Advancement of Sciences and the National Academy of Sciences. And from us, she's received the prestigious Pioneer Award grant, which is given to very few of the most outstanding scientists in the United States. And I think it must make her feel very good to be an honorary professor of the Chinese Academy of Medical Sciences in Peking Union Medical College, her alma mater, and as well as an honorary professor at Zhejiang University, a very prominent university in Zhejiang province. So, Hao, thank you very much for being with us today. On behalf of the biomedical research community, I welcome you to NIH.
Thank you so much, Mike. That was very kind of you. I'm delighted to be here. Actually, I had a great fun talking to some of you this morning, with PIs as well as students and postdocs. So I would like to spend the time today to tell you something that we have been working on for a long time, but now just finally they're coming together, as well as some unpublished data. And guess who I bumped into this morning trying to get breakfast? So here are my disclosures. So let me get right to it. So inflammasomes were first identified. Just want to make sure this mouse works. Yeah. Oh. Is this the pen? This is the pen, okay. Identified as the supramolecular complex that activate caspase 1. And this initially came from York Chop's lab. Yeah, I used to like this one because I can draw. Okay. Oh, okay, I'll try to draw. Yeah, this is a PC, it's not a Mac. Okay. The first inflammasome was actually identified in my friend and collaborator's lab, the late York Chop. As you can see from this beginning, it's already known that inflammasomes contain a protein that's known as the sensor, and then the adapter, and then, of course, caspase 1, because that's the function of inflammasomes, and the fact that they form these large molecular assemblies, as you can see from their migration out of a gel filtration column. So as you can see from this timeline, the inflammasome discovery really was a breakthrough for the field of fever and inflammation because it follows the first discovery of the interleukin-1 cytokine as the fever-producing protein, and then the caspase-1 as the enzyme that cleaves pro-interleukin-1 beta to generate the mature cytokine. So now, of course, inflammasome activate this caspase-1. So we now know that canonical inflammasomes are composed of these supramolecular structures, and I list here some of the sensor proteins that's in these inflammasomes. And in today's talk, I will talk about some of our work on the NLRP3 inflammasome. At least that would be part of the focus of the talk. So these sensor proteins, upon activation by pathogenic or endogenous danger signals, it will recruit this adapter protein and then caspase-1, of course, and this will generate dimerization and auto-processing of caspase-1 to induce inflammasome activation. So the inflammasome itself often forms at a single speck in the cell. So this will come back later because this is a special location in the cell at the microtubule organizing center. So activated caspase-1 or other caspases that are also pro-inflammatory, which are induced to activate through what we call a non-canonical inflammasome pathway, can all cleave this protein known as gasdermin D. Gasdermin D was initially discovered in the lab of Feng Shao and Vishwar Dixit from when they used genetic approaches to try to look for proteins involved in pyroptosis. So it turned out gasdermin D can be cleaved by these caspases, and then the N-terminal region that's released from this cleavage, as we showed and several other labs showed, interact with lipids at the cell membrane and then form pores. And these pores are important for the release of the mature cytokines to the outside of the cell because these mature cytokines do not have any signal sequence, as well as to induce the cell death that's highly inflammatory, known as pyroptosis. So let me start from the NLRP3 inflammasome. So a little bit different from other innate immune sensors. NLRP3 is not a pattern recognition receptor. Instead, it senses something more general. And the emerging concept is that NLRP3 senses some kind of a cell membrane damage. So many different things can activate NLRP3. I highlight a couple here. One is the bacterial toxin nitrorhizin, because it damages the cell membrane to allow potassium efflux, or extracellular ATP, which is a damage signal that can activate a potassium channel. And these particulate structures, aggregate structures in the cell that can each induce or associated with certain important human diseases, such as uric acid for gout, cholesterol crystal for cardiovascular disease, amyloids for neurodegenerative diseases, and even PM 2.5 for environmental lung diseases. So NLRP3, if you look at its domain organization, it has this so-called pyrene domain and the N-terminus. This belongs to what Mike introduced to you as part of the DES domain family. And then it has the so-called NAC domain. This domain essentially is an ATPase, but its function is not to generate a force, for example, due to ATP hydrolysis, but rather by binding to ATP, it somehow induces a aggregation of the protein in order to perform its function. And the leucine-rich repeat domain, of course, is well known for their function in mediating protein-protein interaction. The ASC adapter also has a PYD domain, and the PYD can interact through these filamentous structures that's common to the entire DES domain family. CARD is also a DES domain family member. C then is also able to recruit the caspase molecules through this CARD-CARD interaction, which then bring the caspase together for their activation. So interestingly, in cells, they form these large specks that are essentially large aggregates of different kinds of filaments that we can further represent them or recapitulate them in vitro. So the question for a few years now has been, how do then NLRP3 form the initial oligomerization platform in order to nucleate the filament formation of the PYD and the CARD? And I will go through a journey that we've taken in recent years through 3 different cryo-EM structures, and on top of it, this extensive intracellular trafficking that we discovered along the way. So the first cryo-EM structure that we solved with NLRP3 and NEX7 is the NLRP3 that doesn't have the PYD. We deleted the PYD because we worried that the filament formation ability would make this protein highly aggregate. So we solved this cryo-EM structure in complex with NEX7. What is NEX7? NEX7, in fact, is a centrosomal kinase. It localizes at the centrosome. And the catalytic activity is not required for its supporting of NLRP3 activation, but it is the scaffolding function that is required. So when we solved the structure, we saw that the leucine-rich repeat domain, this is of the NLRP3, directly cradle this NEX7 kinase. So that was all we know at the time. And we do also know that the NLRP3 in this structure is in the ADP bound state. Remember, this is a ATPase, ADP bound state, which is inactive. So then a couple of years ago, we solved the cryo-EM structure of the full-length NLRP3 with the PYD. Even though the PYD is there, it looks like it's completely buried within this, what we call a double ring cage, a cage that has 2 layers, 5 or 6 on the top and 5 or 6 on the bottom, and PYD is hidden inside this cage. And that's probably why that the NLRP3 inflammasome at this stage is still inactive. And the NAC domain, which is shown in this region of the molecule, is still ADP bound, not ATP bound. So what was surprising was that when we looked at these NLRP3 cages, we found that they're mostly membrane bound. So when we fractionate these cells that's overexpressing NLRP3, most of them are actually in the membrane fraction. And if we solubilize the membrane fraction, we get the same kind of cage-like structure that we saw from the soluble fraction. In fact, this can also be recapitulated in macrophages. In this case here, for example, in the wild type immortalized bone marrow-derived macrophages, the membrane fraction has a band that corresponds to the purified, where the purified NLRP3 would have run on the native page, suggesting that these membranes actually containing the cage. So the cage has this interesting structure that this region here, which is the linker between the PYD and the NACH domain, right, right here, this region here, has a lot of positively charged residues. And we show that when you mutate these residues, they actually can no longer be on the membrane, and they also cannot form the cage, suggesting that this binding to the membrane is important for cage formation. So James Chan's lab a few years ago found that NLRP3 stimulation induces Golgi dispersion. So that is the Golgi, which usually is a cap-like structure right next to the nucleus. Upon activation of the NLRP3 inflammasome, the cap completely disappears. It becomes tiny little vesicles. And we subsequently showed that these vesicles actually get trafficked to the mTOR. This is at the mTOR. And this is a TGN marker here. These TGN-containing vesicles, if you will, are trafficked to the mTOR to bring NLRP3 to the mTOR. So remember that NEX7 is a centrosomal kinase. So we'll come back to that in a bit. So then we show that the NLRP3 cage, why do we have the structure, right? It's kind of a complicated structure which prevents NLRP3 activation, but why do we need it for NLRP3 activation? So it turned out the cage itself is important for the dispersion of the TGN in this early event after NLRP3 stimulation. So as you can see here, in the wild type macrophages, these are mouse macrophages, these are the TGN staining. NLRP3 is partially at the TGN already. Upon stimulation with this bacterial toxin nitrorhizin, NLRP3 and the TGN, they all colocalize at the mTOR. And you can see the entire TGN disperses into tiny vesicles. And this dispersion is NLRP3 dependent. If you use NLRP3 knockout cells, you don't get this dispersion upon treatment with the NLRP3 activator. And if you reconstitute these cells with a mutant of NLRP3 that cannot form the cage, you also don't get TGN dispersion, suggesting that the cage is important for this process. So this led us to hypothesis that the cage itself may exist for a reason. It not only suppresses the NLRP3 activation, but is also important for this early step in its activation by dispersing the TGN. So now we kept on solving NLRP structure in the inactive conformation. No matter what we do, everything we purify when we overexpress NLRP3 is in the inactive conformation. So this changed when my postdoc, Le, decided to try something else. What he did was to co-express NLRP3 with NEX7, these are the two important proteins, in a cell that's very similar to HEC293, and then he treated the cells with nitrorhizin, basically treated the cell with the same stimulus that would have activated NLRP3. And then he lysed the cells, and then he add this ATP gamma S, non-hydrolyzable ATP analog, ATP gamma S, to the lysate to stabilize the ATP conformation. And on top of it, he added ASC, because if we don't have all three of them, we do not get this ordered NLRP3 structure. I think partly because the NLRP3 inflammasome, unlike the previous inflammasome structures that we solved, which is NLRC4 inflammasome, in there, ASC was not required for the formation of the complex. But here, you need the ASC to stabilize the NLRP3 complex. So you can only obtain this tertiary complex between NLRP3, NEX7, and ASC. So here we can see the different dislike structures. That's either 11-fold or 10-fold. And the structure with the 10-fold reached the best resolution, and therefore I'm showing you the 10-fold structure. So this is the individual subunit, and in the cryo-EM map, this is where NEX7 is. Sorry, I'm not drawing it well. The yellow is NEX7, and everything else is NLRP3, this whole thing here. And this NACH domain now has changed conformation to an ATP bound conformation. And if you look at the side view, you can see that the added ASC PYD is together with the NLRP3 PYD to form a short filament next to the NLRP3 disk. So just showing you briefly the conformational change that's occurring between the inactive state and the active state. Basically, this LRR domain, as well as part of the NACH domain, they together have this massive movement, almost 90-degree movement. And I won't detail these other changes, but there's also local changes in other important domains in the protein, especially this domain that has been implicated as a sensor for potassium efflux. I think there may be a lot more story behind that, but we currently don't know how this domain or other aspect of the biology senses the NLRP3 activators. So here I just show you that instead of ADP, now we have ATP bound. This is the whole thing here. And this residue here, histidine 522, in the inactive conformation would have been in clash with the extra phosphate group in the ATP, and therefore this would induce a conformational change to the active state when ATP is bound. So one of the most prominent question from these structures, I think, is the fact that NEX7, which is, I showed you this before already, NEX7, which is right here, it doesn't really associate with anything other than the LRR. It doesn't mediate oligomerization. And then the question would be then, what is the function of NEX7? So we hypothesized because NEX7 also binds the leucine-rich repeat domain, and this is the same as for the leucine-rich domain interaction in the cage. So we hypothesized that these two bindings are competing with each other. And indeed, if we start with a cage structure, and then we add NEX7, as shown here, the cage, which is this peak here and this peak here, the cage would be disrupted, and suggesting that indeed NEX7 can disrupt the cage. So this biochemical data, as well as cell biology and structural data, prompt us to propose this hypothesis of trafficking and conformational changes in NLRP3 activation. That is starting from monomeric NLRP3 or the cage form of NLRP3 at the trans-Golgi network. There may be an equilibrium between this monomeric state and the cage state. Upon NLRP3 activation, we imagine that the ATP exchange would have occurred already, so that this NACH domain changes conformation to a more standing up conformation. So the closed conformation is more compact, whereas the active ATP bound conformation is more standing up like this. And then this kind of cage is able to be transported to the mTOR on the microtubule network. This actually been known for quite some time. It's actually one of the first discoveries of inflammasomes is that it is sensitive to microtubule disruptors. And in fact, colchicine, which disrupt the microtubule, is an inflammasome drug. So once it gets trafficked to the mTOR, we hypothesize then NEX7 would be there to disrupt the cage. And because the two layers in the cage structure are already linked together, they're already linked together through the PYDs, one could imagine that this will just unfurl the cage into two 5-subunit sub-complex, and then these two can just come together to form the final disk that contains 10 subunits. And this, of course, need to be further stabilized by the bound ASC, shown here in red. Why NLRP3 use such a complex mechanism for its activation? We think perhaps any inflammasome activation has some levels of terminality. So now, if you induce cell, pyroptosis, cell death, that's the end of the cell. So probably the cell have developed many different regulatory mechanisms in order to suppress its activation until every step is clicked. Every step, every checkpoint is clicked so that NLRP3 can be activated. So interestingly, NLRP3 is conserved throughout revolution. And bat is an interesting case. So the bat is known as a reservoir for many different viruses, as you know, which may include the coronavirus, the pandemic that we have just suffered through. So it turned out bat NLRP3 has different isoforms in which this exon 7 in the leucine-rich repeat is deleted. And if you look at these deleted isoforms, it has, which is isoform 2 and 4, it has very low activity. Isoform 2 here and 4 here have very low activity. So we hypothesize that because of this deletion in the NLRP3 domain, it may still be able to form the cage, but may not be able to bind to NEC7, and therefore you cannot activate the bat NLRP3 inflammasome. And this is something we're testing in our biochemical and structural studies. So another interesting question that we're trying to address is what are the effects of these cryopyrin-associated periodic syndrome mutations, the CAPS mutations. Almost all these mutations are mapped to the NACH domain where the conformational change occur. But we don't know whether this automatic conformational change in these proteins would already cause TGN dispersion, or you still need a second stimulus, maybe a very weak stimulus, in order to activate the pathway. And we thought this could be a very interesting one to address. So as a summary for this part of the talk, basically we showed that the different NLRs, so I talked about the NLRP3 inflammasome, but there are also a lot of other inflammasomes, such as the NLRP6 and NLRP1 inflammasome. What was striking to me was that the different NLR proteins, they all have the same domain organization. They all use very different ways to activate the inflammasome. For example, I didn't talk about NLRP6, but NLRP6 goes into a phase separation in the presence of double-stranded RNA or a bacterial component. And this is the initial signaling platform for the NLRP6 inflammasome activation, whereas NLRP1 use a cleavage in its protein in order to release a toxic inflammasome-forming fragment to induce the activation of this particular NLR inflammasome. So we look forward to learning what other kind of inflammasomes may do in its ability to induce inflammasome formation. So now I'm going to switch gear to talk about gasdermin D. So gasdermin D, as I mentioned earlier, is a substrate of all inflammatory caspases. We actually now know that gasdermin D can also be cleaved by a number of other enzymes. I think I probably have a slide in the next few slides. So we showed early on that gasdermin D binds to acidic lipids. So if you remember, where are the acidic lipids in the cell? Certainly they are localized at the inner leaflet of a mammalian cell membrane. And that's why gasdermin can go to the cell membrane and punch a hole from within. And once they get released from the cell, they actually cannot attack the mammalian cell anymore because there's no acidic lipids. And the fact that it can also interact with cardiolipin, this is a lipid that's present in mitochondria, also suggests that gasdermins can damage the mitochondria, and now there's a lot of evidence supporting that. So here is the gasdermin family. This is a family that has been there for a while, but didn't draw a lot of attention to the field until the discovery that the N-terminal domain can induce pyroptosis. And it turned out all the different gasdermin family members from A to F, if you will, this is a F member, A to F, its intermingle domain can all induce pore formation and pyroptosis, and they can be activated by different kinds of enzymes. I list a few on the side of this slide. I don't know if this, okay. So why pyroptosis? Pyroptosis is very different from apoptosis because it can damage the mitochondria so that things can go out, things can also go in. So ATP can go out, of course, lactate dehydrogenase, HMG B1, a lot of different danger molecules can go out. So they're very immunologically hot. By contrast, apoptosis is immunologically cold, so they don't really alert the immune system to attract the attention there. So over the years, we've solved 2 different gasdermin NT intermingle domain pore structures. I just want to draw your attention that gasdermin pores may be formed through two different steps. One is the formation of a prepore. So what is a prepore? So this is the yellow here is a prepore structure. So the prepore is also already oligomerized on the membrane, but it has an insert into the membrane. So as you can see here, the inserted part of the structure is a beta barrel. This is a large beta barrel that contains a lot, 33 times 4 different number of beta strands. A lot of beta strands that makes this large beta barrel. So the prepore can be formed, and then insertion can occur in order to form the pore structure. This will become important when I talk about the remaining part of the talk. Some of it unpublished. So the subunit of gasdermin looks really like a left hand. This is the inactive structure like this. This is like the globular domain. And during insertion, you just have your four fingers as the two pairs of beta hairpins that goes into the membrane. Once you insert your fingers like this, the pore is formed, and the pore becomes this transmembrane pore that can release the cytokine or other molecules. So this is a simulation of the refolding events in this pre-pore to pore transition. This yellow starts with a pre-pore, and then the strands insert into a pore. So once we solved the gasdermin D pore structure, there was one question that bugged us, that is this paradox. Pro-interleukin-1, which is also very small, it's about 31 kilodalton, cannot be released from live cells. So these live cells can still have gasdermin D pores on the surface, and they can release mature interleukin-1, which is 18 kilodalton, but they cannot release the pro-cytokine. And these are the data that showed here. So the pro does not come out, but the mature cytokines come out. Size-wise, they are about, in terms of diameter, a quarter or less of the big pore structure, just like this size here for a big pore, but it cannot come out. So a prominent feature of the pro-cytokine that's absent in the mature cytokine is this negative charge. So the propeptide here is actually extremely negatively charged. So we thought, is it possible there's some kind of a charge selection for the gasdermin D pore selectivity? So gasdermin D, if you look at the isoelectric point, or the PI of gasdermin D, is actually 8 point something. So you would, at least I did not, I would never thought this could be have any negative charge in this protein. But it turned out, even though the PI of the protein is very high, if you look at the charge distribution on the entire pore, all the strong positive charges are facing the membrane, and that's because they're binding acidic lipids. And inside the pore, you see inside the pore is actually very acidic. And when you calculate the electrostatic potential of the wild type versus mutants, AP1, acidic patch 1, or acidic patch 2 and 3, when you calculate the electrostatic potential, you do see that when you mutate these residues in silico, you have reduced amount of acidic electrostatic potential. So we did a lot of different tests in vitro, but I just want to show you this last test that we did in cells, in which we used gasdermin knockout cells and then reconstituted with either the wild type, sorry, I'm so terrible drawing there, wild type or the acidic patch mutant of gasdermin D, and then stimulated cells with very low dose nitroricin for a long time, which the cells do not die, and look at the release of interleukin-1 versus the pro-interleukin-1. So as you can see here, with the wild type reconstitution, you have only the mature cytokine coming out very nicely. Whereas for the cells that's reconstituted with this acidic patch mutant, you see pro-interleukin-1 already coming out, and you only have a little bit of the mature interleukin-1 in the media, and that's because the pro-cytokines are already out so that they didn't have a chance to be processed. So really suggesting that this selectivity for the release of mature cytokine, and perhaps also a lot of other things, is governed by this charge distribution, what we call electrostatic filtering in the gasdermin D pore cargo release. Why does the pore have this selectivity? Is it really evolved only for interleukin-1 release, or is it also important for some other aspect of the biology remains unclear. But one discussion that I had, I think with Ruslan, in fact, Mesitov, he said a lot of danger molecules are actually positively charged, which means they can be released from the pore. So we shall see if that's the case when we perform the secretome experiment, which is ongoing. So I'm just looking at the time. Okay, so in the last part, I want to tell you an unpublished story on this question that has bugged us for a while. So we know back in 2016 when we discovered that gasdermins can form pores, we mutated all the cysteine residues on gasdermins and found that cysteine 191A is defective. If you transfect the N-terminal domain of this mutant, you do not kill cells. But we didn't understand why. And even after we had the structure, we know that this residue is at the tip of the beta hairpin, right at the tip. But there's no disulfide, and these cysteine residues are very far apart from each other in the pore. We don't know what is the function of this thing. So given that this is a membrane protein, I actually suspected maybe it is lipidated. But we really didn't go after it until a brave postdoc decided to try that experimentally. So indeed, that turned out to be the case. The cysteine residue is palmotylated. So palmotylation is one of the lipidation form, but palmotylation is, I believe, the only lipid modification that is reversible, can be regulated. So we used a number of methods to look at this palmitoyl modification on gasdermin D. One of them is called acyl biotin exchange, in which basically you modify all the free cysteines in the cell using maleimide. Then you cleave this thioester bond. So palmotylation is linked together with the cysteine residues through a thioester bond using this thing called hydroxylamine. So, and then after you treat these cells with hydroxylamine, you leave the palmotylated group with a free thiol, and then you can put in anything you want, a biotin that has a free thiol, and then you can pull it down. So as you can see here, full length is palmotylated, N-terminus is palmitoylated, but not the C-terminus. C-terminus is not palmitoylated. I'm not showing you the data, but other gasdermins are also palmitoylated. So we start to think that maybe this is a general phenomena for the entire gasdermin family. So interestingly, if you take a THP1 like macrophage, these are differentiated THP1 cells, you prime the cell with lipopolysaccharide, LPS, you see a little bit of increase in palmitylation. Without LPS, you see nothing. No, nothing can be detected. And then when you treat the cells with LPS plus nitroglycerin, you see a greatly increased amount of palmitylation, both on the full length and also on the N-terminus. Whereas at least under this condition, there's no change in gasdermin deprocessing, suggesting that the entire upstream events were not affected, only this downstream event on the lipid modification of gasdermin D. So we mapped which residue. Of course, you know the answer already. It's C191. I'll come back to that, but this residue turned out to be a really interesting residue. It's involved in so many mysteries about this protein. So we then validate this in vitro to see whether the palmitoylated gasdermin D is more active. So here, this is a liposome leakage assay using purified proteins. So if you purify these proteins from bacteria, I believe the orange here is the wild type from bacteria. The green is the C191A mutant from bacteria. In bacteria, that means they're not palmitoylated. You can see this is the rate of liposome leakage, or this is the rate that these proteins damage the liposome to release the dye. But if you use mammalian purified gasdermin D, this gasdermin D is about 50% palmitoylated, not fully palmitoylated, about 50%. You can already see this about tenfold increase in the rate of palmitylation, suggesting indeed this palmitoyl group really helped to insert gasdermin D into the membrane. So functionally, the same thing, the C191A mutant is defective. And when we looked at them by cellular imaging, wild type can go to the cell surface. You can see form these punctate structures that's on the membrane, whereas the mutant, the C191A mutant, is not really membrane localized at all. So it's affecting membrane localization and perhaps also pore formation. So this I think is where it gets really interesting. So because this is a thioester bond, so we had worked with thioester bond before, and we know that any reducing agent, anything that has a free thiol, can attack this and destroy this thioester. So we thought, this probably means that this thioester is sensitive to the redox state of a cell. If you have more reducing agent, this will not stay. If you have a more oxidizing environment, maybe this will stay and stay longer. So then we thought, okay, maybe then this will link it to the generation of RLS, which changes the redox state of the cell. So what we did then was to use the same THP1 cells, but also instead of using just LPS plus nitroglycerin, we also treat the cells with rotenone is one, antimycin A. These two are inhibitors of the mitochondria respiratory complex, complex 1 versus complex 3, and they all induce RLS in the mitochondria. So these two, as you can see here, increases palmodulation, but no change in the cleavage. If you treat the cells by contrast using N-acetylcysteine, this is basically a reducing agent, you reduce the amount of palmodulation relative to non-treated. And this is another inhibitor that also reduces the cellular RLS. So So this is already saying that, okay, if you have a RLS enhancer, you have more palmotylation. If you have a RLS inhibitor, you have less palmotylation. And what happened to the cell under those conditions? Are the RLS really increased or decreased? So this is the condition where you treat the cells with LPS plus nitroricin. This is by imaging the amount of cellular RLS that you observe, or the mitochondrial RLS you observe. If you treat the cells also with this 2-bromopalmitate, which is an inhibitor of palmitoyltransferases, you reduce the RLS. And if you treat the cells with the one that increase palmotylation, you also greatly increase the palmotylation, you also greatly increase the RLS, suggesting that RLS and palmotylation are really coupled. They're together. One enhances the other. So then we looked at among the 23 human palmitoyltransferases, and they all named this guy here because they have a D and 2H and the cysteine at the active site. So we tested all of them and found several of them may enhance the palmotylation of GSDMD. And judging from the expression pattern that we obtained from the Protein Atlas website, we chose to investigate further these 5 different palmitoyltransferases. So when we knock down one by one these different palmitoyltransferases, you see that 5 is very important for the palmotylation, and 9 is kind of important as well. Similarly, the knockdown of these transferases also reduce cell death, membrane localization, and everything else. So, so this then led to our model about what RLS and palmotylation may together regulate GSDMD activation. So this 5, ZD, HHC5, or 9, have all been shown to localize around the Golgi and maybe also the ER, or maybe also other cytosolic locations. So we think that when GSDMDs are not cleaved, maybe the main source of palmotylation is through these cytosolic ones. And when you have higher RLS, you get more of palmotylation. So this little squiggle here is the lipid. And then GSDMD, either palmotylated or not palmotylated, can be activated or cleaved by mature caspase 1 or other mature inflammatory caspases. And if the cleaved GSDMD is already palmotylated, it will go to the membrane, form a prepore, and then immediately transit into the pore itself. So why do I say that? I think just structurally, if you look at the prepore, and if you look at the location of where palmotylation is, right at the tip of the inserted bits, the lipid that's attached to this fingertip here, I imagine at least may help to drag the insertion process so that everything will get inserted into the pore. Another reason I said this is because John Kagan's group published a paper just maybe about a year ago, and they showed that this mTOR-related RLS is important for the insertion of the pore, but not for the localization per se. So this, I think, at least mechanistically can explain that. RLS, which affects some membrane localization and the formation of the prepore, but maybe the most important effect of this is to drag the prepore into the pore to form this functioning pore. So once it's cleaved, if the prepore can also be formed on the cell membrane, then this cell membrane form of palmitoyltransferase. So only 5. Actually, among all the different palmitoyltransferases, I think 5 is the only one that's known to be on the cell surface as well. So I would imagine the 5 would be the major one that would change the pre-pore to a palmotylated pre-pore and thus facilitate its insertion. I'm just looking at my time. So in theory, it has been shown that palmotylated stuff, when they get into the membrane, they tend to cluster. And this is because these lipids, palmitate, is aliphatic. They don't have any double bond. It's aliphatic. So they're very ordered. They can cause phase separation in the membrane so that the pores or the palmotylated groups now become phase separated into a clustered entity with multiple pores altogether. And this is indeed what we saw on the light imaging data. We don't actually see a even distribution of gasdermin on the cell surface. We see these punctate-like structures. So I imagine that this will also cause a phase transition and clustering of the pores by the palmotylation effect. So interestingly, a few years ago, we published this disulfiram, which is a thiol modifier. Guess what? They all modify cysteine 191. And Abbott's group and Kate Fitzgerald's group also showed these other molecules, and they all modify cysteine 191, and they all inhibit gasdermin D. So we're proposing that these gasdermin D inhibitors target the same residue in cells and inhibit their function by competing with palmotylation. And fumarate, so dimethyl fumarate works, but fumarate also works. And fumarate is a molecule that gets upregulated during inflammasome activation. So this then may also act as a negative feedback within the cell to suppress further inflammasome activation after the occurrence of inflammasome activation. So as a summary, I showed you some data on our NLRP3 project, suggesting that NLRP3 is activated through not only large conformational changes that's associated with ADP versus ATP binding, but also extensive intracellular trafficking. And perhaps these steps can be targeted by therapeutics. At the moment, all the NLRP3 targeting drugs are targeting the ADP form to keep it in the ADP form so that it doesn't get activated. And then we showed gasdermin D pore, even though it's so large, we were pretty surprised that it actually has some selectivity. Despite its size, it does have certain selectivity, but we don't fully understand why it has this selectivity. What else does it mediate inside the cell? And finally, our unpublished data showing that gasdermin D is also modified by this lipid group. And I think this connection to RLS also says that pyroptosis is not just any form of cell death. It's a cell death of the last resort, if you will. The cells are already really stressed out by stress, by oxidative stress, for example, and then, pyroptosis is achieved through this conduit. So maybe pyroptosis should really be a oxidative stress-induced cell death. With that, let me thank the wonderful postdoc and students in my lab who performed the work. And I want to highlight my collaboration with my colleague, Judy Lieberman. And I thank you for your attention.
Hello. Wow. Thank you very much.
Thank you.
Let's have another round of applause for
Oh, my God. Okay. Thank you.
That's great. So I didn't get any questions on here, but I'm happy to take, these are for the people from NIH. Oh, wait, there's one here from Jay Chung. So let's do him.
Okay, Jay Chung.
Jay Chung. Some reports have indicated that mitochondrial DNA release is required
Yeah.
for NLRP3 activation. Do you see evidence for this?
Yeah, it's something that we would like to test. I know I talked to Michael Karen about this
Okay.
pretty extensively. Yeah. We tested previously to use our inactive NLRP3 to see if it binds to mitochondrial DNA. We did not see interaction in our hands. But now we also have the active state. We're going to test again and see if it's the active state that the mitochondrial DNA targets. Yeah, I would love to make a connection between the mitochondrial pathway and the TGN. I don't know how to rationalize this whole thing yet, but hopefully we'll get there.
Okay,
Yeah.
great. Other questions? Yes.
Very impressive. This is Peter.
Thank you.
Work. So you described extensively of NLRP3 and its role in the vesicle trafficking and TGN stuff, M2C. But the NLRP, the primary function is activate procaspase, right?
Yes.
So do you see any evidence how procaspase 1 interact with the inflammasome and
Yeah.
then subsequently?
Yeah. So that actually, I think that question, well, it may be more complex, but in our, I think 2016 paper or something, so we looked at how the CAR domains interact. So procaspase has a CAR domain at the N-terminus. It interacts with the ASC CAR domain. And once this interaction occurs, the caspase catalytic domain would be brought together, and then they will dimerize so that they can then auto-process, trans-auto-process to become cleaved and active caspase 1. So in my head, this is what's happening. But I think the story probably is much more complex. We have some unpublished data, which is still not completely clear. But we think that the direct recruitment of pro-cytokines, L18 or L1, is not that simple. It's actually, it's more complex than just a simple recruitment. We think there's more steps that's involved in that, but we don't know about the substrate recognition yet. But I think we're pretty clear about the caspase activation, but we don't know about substrate recruitment. And as you know, Feng Xiao's group and also I think Sam Xiao's group have shown that gasdermin D needs this exosite to be recruited to active caspase 1. So it's not just the active site interaction, but also an exosite interaction. In fact, this interaction is more important than the tetrapeptide motif recognition. So I think our data maybe begin to show that the some kind of an exosite interaction is also for pro-cytokine processing. Yeah.
But my understanding is that activation caspase also require assembly of inflammasomes. So is it possible to put a catalytically dead caspase construct into your?
Yes, absolutely. Yeah, absolutely. Yeah. Mm-hmm. We think dimerization will still occur within the filament. Yeah.
Yes.
Oh, thank you so much for a wonderful talk, Dr. Wu. My name is Won-Young Lee from Dan Kastner Lab.
Okay, cool.
I am actually fascinated about the palmitoylation modification of gasdermin D, and your data beautifully explains what happens when an NLRP3 inflammasome is activated. I'd like to ask, in case other inflammasomes are activated, do you think the same thing occurs to gasdermin D in terms of palmitoylation? Because
we haven't tested, but I think so. I think because this is a physical principle, so I don't think it'll be different unless there's other mechanisms to compensate for that. I imagine all inflammasome activation somehow raise ROS. I don't know how, but somehow raise ROS, and that will cause gasdermin D palmitoylation and help to induce pore formation. That would be pyrene, for example. Yeah. At least that's what I imagine. But we haven't tested that.
Okay.
Yeah.
Another question is, so do you think that in other gasdermins, is the cysteine residue conserved?
Yeah.
And then
No, the cysteine residue is not conserved.
Oh.
But all the gasdermines that we've able to test so far are pore modulated. There's only one we couldn't test because it didn't express, which is gasdermin C. But everything else we tested are pore modulated. As a structuralist, I like simple things, so I think there got to be some commonality among the different gasdermins.
Okay, thank you.
Yeah.
Yes, another question.
Very nice talk. With the NLRP3 summary slide, you showed that the inhibited but caged form is what gets trafficked to the MTOC. Just like, how does that happen? Why does it have to be in that caged, inhibited form? How is that selectivity enforced?
Yeah. We're trying to figure that out. We don't know. So I think the cage is required for the Golgi binding because you have multivalency. If you just have one, positively charged patch, it's probably not enough. You need the whole thing. So, but why does the cage induce Golgi dispersion? That we don't know. Then that's a question we really want to address. Yeah.
You could.
My name is Kun. I'm a postdoc in Max Lab.
Okay.
So I'm wondering if gasdermin D is only selective for cell memory or like other also for other
other
memory?
membranes too. I didn't mention that, but certainly mitochondria. Certainly mitochondria.
Only for two-layer memory?
Yeah, so there's, maybe those are not published, but I think mitochondrial damage by gasdermin has been implicated. Maybe not directly shown, but implicated, yeah. So the idea is that the cardiolipin that's in the inner membrane of the mitochondria can be externalized to the outside. In fact, the scramblase that's important for this process is important for gasdermin D localization to the mitochondria.
Okay, thank you.
Does it make sense? Yeah.
Another question is, as you said, the gasdermin D we cleaved into a C-terminal and an N-terminal. So what's the function for C-terminal?
Yeah, I have no idea what the C-terminal does. Yeah, I have no idea. Yeah,
Okay.
yeah.
Do they form like the dimer or only the C-terminal release and functional as a...
Yeah, we don't. So the only function I know about for the C-terminus is to suppress the N-terminus for sure, right?
Yeah.
Because in the full-length protein, it does that. Once it's released, what does it do? I have no clue. I have no idea what it does afterwards. Yeah, maybe it's not fully. So I want to mention one thing. So gasdermin D, I didn't talk about that part of our work, but gasdermin D is expressed widely in cells, not just in macrophages. So that was really strange. If it's downstream of inflammasome, why is it expressed in other cells? For example, in a lot of cancer cells, it's expressed. So I don't know, but I imagine gasdermin D has other functions. Maybe that will have to do with the C-terminal domain. It's anyone's guess, I think, at this point.
Okay.
Yeah.
So the endem cleave, the gasdermin D is only caspase 1 or 11?
No, there's also other ones.
Or like grandam, some...
Granzyme, which one? B, I think. No, that's cleaving E. That's actually cleaving E. It's better I go to my talk. I actually don't remember. Let's see. I don't know how to go back. Okay. So, yeah, it's here. It's here. It's this one. D is, right, caspase A also cleaves D. Neutrophil elastase also cleaves D. And the neutrophil elastase cleavage of D is actually important for netosis. This neutrophil exclusion of nuclear DNA. And caspase A, through the apoptotic pathway, can also cleave gasdermin D.
Thank you.
Yeah.
What is the length of this hydrophobic domain?
Yeah, it's about, I measured this before. Now I don't know if I remember. When I was writing the first paper, I measured it. It's about a slightly compressed double layer. It's like slightly compressed, I would say. So it's possible it causes some kind of membrane curvature as well.
Now, if you
It is going to the plasma membrane already. Yeah, if you change the hydrophobic bits, yeah, and not done that, yeah, you wonder whether that will change the selectivity. Yeah, it may. Yeah.
Yeah, one final question.
Okay, thank you for the talk. I have one question. Why the NLRP3 inflammasome assembled at the MTOC microtubule? I want to know the molecular basis or, or the physiological function.
Yeah. So we found the reason that we investigated was because we see these single specks in the cell. We were like, why? Why is it only one? Why is everything coalesced to one? So, and it's perinuclear. So we were just like, it looks like mTOR, because that's where mTOR will also localize to. So we tested by immunofluorescence. Indeed, they're on the mTOR. And then this together, and then we tested microtubule sensitivity. Are these things sensitive to microtubule disruptors? Actually, so this is also before us. Other people already, even York, I think, York Chop, already tested colchicine and cortisol, and they all disrupt NLRP3 activation. So we retested those. We also mapped the dyning inhibitor, also inhibit NLRP3, and the dyning adapter, which is HDAC6, which binds to ubiquitin, also inhibit this microtubule transport, suggesting indeed NLRP3 has to be transported there. What's the reason for that? Good question. So my rationale for that is that this pathway is also similar to the so-called agrosome pathway in the cell. So aggregated cellular proteins are usually ubiquitinated, but they're too big, I think, to be degraded by the proteasome. So they get transported to the mTOR, and at the mTOR, there's usually a lot of donor membranes. You know, like you have mitochondria right there, there are lots of things there. So you form autophagosomes. So that's how you degrade these proteins. So in our case, we also saw the spec. This was old work. In our spec, they're enclosed oftentimes by double membranes, autophagosomal membranes. So we think it goes there to activate. It also goes there to be regulated by autophagy. In fact, if we treat the cells with 3-MA that inhibit the autophagy, the puncta goes from here to here, like huge. They just have these gigantic puncta, suggesting that autophagy at least sets a rheostat in the level of inflammasome activation in the cell. So we think we go there to activate and go there to be regulated.
Yes, thank you. I think usually there should be a mechanism recruiting, like the NLRP3 or the inflammasome to the MTOC. I think there should be a mechanism there. So,
yeah, we think it's on the vesicle. That's what we think. The Golgi vesicles have NLRP3, right? The TGN dispersion. We think it's the vesicles that gets transported, but we're not 100% sure. The reason we said it's the vesicle that gets transported is because TGN markers become on the mTOR when you have inflammasome. So we figure that must be the vesicle that gets transported.
thank you.
Right.
Okay, well, thank you very much. That was really, really fantastic. Appreciate it.
Thank you.